Hey, and welcome. I'm Fiona Mischel, and this is the Built with Biology podcast, where I take a deep dive with scientists, innovators, and thought leaders to ask one big question. How can we use biology to build a better world? Hi, everyone, and welcome to the very first episode of the Built with Biology podcast. I'm your host, Fiona Mischel, and as it's our first episode, we're going to dive into one of the most relevant and critical topics in biology today, biosecurity. I am so excited now to welcome James Diggans, the head of biosecurity at Twist Bioscience, a company that is actually making DNA. James, it's just such a pleasure to have you on the show. Thanks so much for joining us.
Thanks, Fiona. I'm excited to be here.
So we're about to dive into this huge topic of biosecurity, but before we do that, can you give us a bit of an overview of, what is DNA? And, as Twist is a DNA synthesis company, how do you actually make DNA?
Sure. So making DNA from scratch is not really new technology at all. So we've been using chemistry to build strands of DNA since the 80s. That process adds DNA bases. You may have heard the A's, T's, G's, and C's. One at a time, like beads on a string. You sort of form molecules of up to maybe a few hundred or so letters at a time. So before Twist, that process was carried out in wells in a plastic plate about the size of an index card. And you could make, one gene from that plate. Twist technology miniaturized that process. So we grow these DNA strands up from a silicon surface. And in that same form factor as these older plastic plates, Twist can make, instead of one gene, 10,000 genes. And we use 99.8% less reagent and chemicals. So that brings down the cost of making that DNA substantially, doesn't compromise quality. That cheaper DNA means that scientists can carry out more and more experiments, they can learn faster, they can really push the science forward.
So this is pretty amazing what you've just described, that, we used to only be able to make one gene per plate, and now you're talking about 10,000 genes per plate, which is just an amazing leap in what we're able to do. So who uses DNA and how do they use it?
A lot of our customers, as you might imagine, are research scientists. So they're using DNA to kind of help them carry out experiments and solve, biological mysteries that they're studying. So things like, how do cells talk to one another? How does the immune system keep us safe, which is certainly relevant these days.
Oh my gosh.
A second group of customers, right, will work on what we might call advanced biomanufacturing. So that's using biological systems as kind of miniature factories. So you can think of like kind of the chemical industry as using oil or petrochemical byproducts as inputs, going into big factories, operating at high temperatures. Those same chemicals that they're producing can be instead made in a large tank of yeast, operated at much lower temperatures, and using sugar as the input instead of fossil fuels. And so it's the DNA really that teaches those yeast what to make and how.
Just want to make sure I get this right. So the DNA is basically the instructions for these little microbes that you put in a bioreactor. You're like, hey, little microbe, please make this particular material. And the DNA is the kind of that core instruction. Am I getting that right?
That's right. Yeah. And please make it this fast. And here's how much to make. And here's how to get along with all the other yeast that are in the tank. Yeah, it's a complicated art.
I didn't realize there was some marriage counseling that goes in that little, no, don't get mad at each other. Don't get mad at each other. I kind of like
that.
It's
like a big playground.
Oh, I'm just like little swing set microbes. I'm going down a tangent here. But so there's that, but there's also, I know that there's DNA data storage. And I want to touch on that very briefly because it is so cool.
Yeah, this is a kind of an emerging use case for DNA. So this is taking advantage of the fact that DNA is a really stable molecule. And nature, of course, has chosen DNA as its primary molecule for long-term data storage. We can take the ones and zeros that are stored on a computer, convert those to the A's, G's, T's, and C's of DNA, store that, and be confident that we could fully recover that data in 10 years, 100 years, even 1,000 years, which no form of magnetic storage can really touch today.
Wow. So all of this is an awesome backdrop, but it's not what you actually do because you're the head of biosecurity. So what does that actually mean? What does your day-to-day job look like?
So my job has two primary parts. The first of those is really operationally focused. So my team and I build and operate software systems to basically evaluate the sequences that Twist is asked to make. And the real challenge there is to determine if those sequences could somehow be dangerous or be misused. And customers do order these sequences. And for those customers, I work with our customer support group to follow up with the customer, have a conversation with them, verify that this is a legitimate use for that sequence. And if there are any licenses or other documentation required, that we make sure that those are in place. And you can think of an exaggerated example, which are always fun. Let's say a customer from the Centers for Disease Control orders a virus sequence from the Ebola virus. So we can be confident, after having a conversation with them, that they have appropriate training, facilities, work history, and we can safely ship them that DNA. But if the same sequence was ordered from a PO box in North Korea, we wouldn't accept that order.
Has that happened? Have you gotten PO box from someone's basement in a country that's maybe a little bit concerning? Has that actually happened? I don't know if you can answer that.
I mean, not from North Korea, but yeah, we certainly do sometimes get strange addresses, but generally they turn out to be harmless. We do engage carefully with those customers and request extensive documentation to make sure that they are who they say they are, and we'll do with the material what we think they will do with it.
Okay, so that's the first part of your job, which is a bit more inward focused in terms of Twist, the company, what you produce, checking on these, possibly suspicious PO boxes. But there's also a more outward facing part of your job, right?
The second piece is really outreach. So the science here is moving really fast. There are lots of stakeholders that want to know about how, risk evaluation and that sort of thing can work. So we, lend Twist expertise to help the US, other governments, other organizations kind of think through the security concerns as these synthetic biology techniques mature. So scientists and policymakers, they're all really good at kind of emphasizing the positive aspects of new technologies. We're all very excited about that. But it's really critical to ensure those technologies are used responsibly. And that really requires having a security-focused view from the outset. And that's something Twist can help them understand.
Yeah, I think you touched on something that's so important. It's like the hype of the science itself. The science is amazing, but not talking about the downsides, not talking about the potential risks, that's a huge disservice to not just the industry, but I think to the, obviously to the general public. And so I want to break this down even a little bit further, which is, what's the difference between biosecurity and how does that differ from biosafety?
Yeah, and that's a really important question. So biosafety you can think of as kind of a set of practices and protocols that are really focused on protecting individuals working in a lab with biological organisms. So those practices might govern what kind of gloves or masks to wear, or how to decontaminate a work surface once you finish an experiment. And importantly, those measures these biosafety measures apply to well-trained individuals. They're doing work they're explicitly authorized to carry out. Biosecurity, in contrast, is really focused on preventing intentional misuse of biological materials to cause some form of harm. So it's trying to answer two questions. Can this biological material that I'm thinking about be misused? And if so, how do we ensure that material is only provided to individuals who use it responsibly? And Twist as a company invests really heavily in both biosafety and biosecurity. Both are important. So we want to make sure that we keep our employees and our customers safe, and that's biosafety. And we also want to make sure that our products are used only in responsible research, and that's biosecurity. And that's where I focus.
Yeah, thank you for that breakdown. That really helps clarify because, some of these sequences, are they toxic? I think that's my next question is, does synthesizing DNA pose a risk to human health or the environment for that matter?
And you can think about that kind of in two ways. So some DNA sequences do code for toxins, and those proteins can be toxic themselves. That's definitely a issue of potential misuse, and you have to be careful who you provide that to. The second way you can think about that is, does the chemical synthesis of DNA itself, really pose some kind of risk? And it does, in some cases, rely on compounds that can be harmful. But I think the beauty of Twist technology is really carrying out that synthesis on silicon, miniaturizing the reactions. We use far less of those dangerous chemicals. And in some cases, it's up to 1 million-fold less than traditional DNA synthesis companies. So yes, that helps us lower our price, which is great. But at the same time, it significantly lowers the environmental impact of DNA synthesis. So you get kind of the benefit of DNA without the concomitant harm of having to use such high volume of chemicals.
Yeah, I think that's a key point, though. because as we move into these sort of bio-based materials, like you were talking about, this biomanufacturing, all these things that, are supposed to be better for the planet and not using petrochemicals like you were talking about, the last thing that we want to do is add different kinds of chemicals back into the environment, which are toxic, right? It's like completely defeats the purpose. So one of the things I keep going back to this idea of this accessibility, like you've been talking about, like huge volumes that you can create, and then the biosecurity. So how can we navigate kind of that dichotomy of making DNA synthesis technology so accessible versus the potential biosecurity risks?
Yeah, and unfortunately, biosecurity in the context of DNA synthesis is not cheap. So it takes significant expertise, consistent investment, and that fact runs this risk of creating, what you might think of as a perverse economic incentive to lower your operating costs by kind of underfunding or ignoring biosecurity practices altogether. And at the same time, the technology is moving so fast that governments are, perhaps appropriately, they're hesitant to really try and directly regulate DNA synthesis or other similar technologies. They don't want to risk harming the ability of companies to compete, because DNA synthesis and synthetic biology is a very international market. So you want to keep companies competitive. Governments could, actively incentivize DNA synthesis providers to invest in biosecurity. So you could, for example, require government-funded research efforts to buy DNA only from companies with these, appropriately sophisticated biosecurity screening capabilities. The state of California recently passed a draft bill taking steps in this direction. That's the first major government step that we've seen. But that's a really good way to incentivize companies to invest in biosecurity. But I think Twist provides maybe the right model for navigating that dichotomy. We see biosecurity as an ethical responsibility. So you have this amazing technology, you have the responsibility to make sure that it's used appropriately. And so we treat the cost of biosecurity as just one more cost of doing this kind of business. And resulting from that is this comprehensive biosecurity program, adheres to existing national regulations, adheres to guidance from the US government, but goes really well beyond that and includes investing in innovation and improved screening methods and scalability of screening to make sure that it can keep up with the increasing demand for DNA synthesis.
Yeah, I think that's such an interesting point. I didn't know this at all, that it's not only is biosecurity, I think, complex with all the different variables, but that it's very expensive. And that's, something that I want to ask is, besides government intervention, and is there ways to streamline biosecurity itself? Is that to bring down those costs and make it easier maybe for smaller companies to make sure that they also adhere to those standards? Is that something that you guys are thinking about and are maybe, trying to help other companies achieve?
Yeah, a lot of the focus on our outreach has been to try and talk about ways that this could happen. One of the major things holding back the reduction in cost of screening is really high quality, publicly available annotation of which genes have, harmful biological functions. That there is no list of that, and it makes it very difficult. It means in practice, what we have to do is compare the thing we're asked to make to everything known, and then provide that list of here's what it's similar to someone who generally has a PhD and can interpret what that means. And that whole process is extremely expensive and could be streamlined, heavily. There's a group called the Nuclear Threat Initiative who's working on what they call a common mechanism for screening that's trying to bring that cost down, trying to simplify it so that you could have many more companies who are doing DNA synthesis have access to, at least some basal level of capability.
That's wild. I mean, the fact that there's no database of these genes are not good for our health. So that kind of blows my mind. So I want to talk more about this, and especially in the context of COVID-19. But for right now, we're going to take a break and stick around. We'll be right back with you.
Symbio Beta's Global Bioeconomy Conference will once again be live and in person. After 18 months of digital meetings, Symbio Beta will once again provide 3 days of strategic networking opportunities for those currently involved in the bioeconomy and serve as a field trip to the future for those still defining their biostrategy. The bioeconomy is already booming, but just getting started. We look forward to having you join us to celebrate the amazing progress we've already made and vision cast for the very bright future that will be built with biology. Join us October 4th through the 6th at the Oakland Marriott City Center. For more information, go to symbiobeta.com.
All right, so we are talking with James Diggans, the head of biosecurity at Twist Bioscience. And the earlier part of this podcast, we were really talking about, what DNA is, how it's, synthesized, who uses it, and some of the potential risks in that area. And now I want to talk about this in the context of the COVID-19 pandemic because, I think that put biosecurity on the map for a lot of people. And James, I want to ask you, has biosecurity changed because of the pandemic? Has it changed your current approaches to continued research in infectious disease? Or, what are some of the things that you're seeing?
I think COVID-19 has a really profound impact on biosecurity. And there are two specific ways that I can think of. First, an important part of our screening process for customers who order these sort of controlled DNA sequences that might be potentially misused is to verify that they have past published work or marketed products that are in line with the sequences they're ordering. So you can imagine a lab orders, bird flu from us. We're going to check to make sure that they have a history of publishing in bird flu or that they have marketed products that look for diagnosing bird, some kind of relation to that disease. COVID made this extremely complicated. So the entire scientific world all at once pivoted to study the virus, whether or not their past work was focused on coronaviruses or virology at all. So in that sense, COVID made this part of our screening process way more complicated. So we had to have a lot more conversations with customers. We had to make sure that we could trust that they intended to use the DNA that we were providing for responsible research. So that was a challenge.
This is an entire system that what you're describing got completely thrown up in the air because the entire world pivoted to focus on one problem, and all of a sudden you can't actually tell who's legitimately working on it and who is like, well, again, that PO box in North Korea. So, you have so much experience in this. How was that for you?
It was really fascinating because at the same time that everyone was pivoting, we also didn't know much about the virus. So the risk evaluation about how dangerous is the sequence of SARS-CoV-2 was an open question, and it wasn't regulated, by the U.S. government at least for export. No licenses required. So, the access to it is technically unregulated. So it was really the responsibility falls on the DNA synthesis organization to make sure that we do the proper due diligence to ensure that we trust the person that we're sending this to is going to use it properly. So it really just required a lot more investment in time, human capital to make sure that we were doing the sufficient due diligence.
How do you do that though? I mean, is this we call it biosurveillance, I guess, of how do you actually find out, if you can't use past published papers and everything's happening so fast, it's a completely novel virus, unregulated by governments. How do you actually go about in determining who's a bad actor and who's not?
Yeah, this is an incredibly hard problem. And there's no perfect answer to it. I think the primary method that we use, which is recommended in a 2010 guidance document from the US government to the DNA synthesis industry, is to follow up with the customer and just say, you ordered this sequence from us. What are you planning on doing with it? And have them provide a response. And if that response makes sense biologically, if they clearly know what they're talking about, the response seems appropriate, the institution they work on has the right facilities, maybe they don't work on coronaviruses historically, but it's a virology institute and they're just pivoting to look at a different kind of virus. It's fantastic. That's fairly low risk. But it is, there's some art to it, and it's not a perfect science.
I do want to talk a little bit more about not just the biological biosecurity risk, but also the AI part of this, because DNA synthesis and other bioengineering processes are relying more and more on artificial intelligence and machine learning to create these products that we've been talking about. So how does that fit into this biosecurity picture? How does Twist actually use machine learning and AI, and could those also pose weak points for biosecurity other than the sequences themselves?
Yeah, I think like most businesses these days, so Twist works very hard to try and squeeze as much value out of the data we generate as we can. And that includes building predictive models on that data. So as a good example, when a customer submits a sequence for us to manufacture, we have a model that kind of estimates how easy that sequence might be to manufacture and then recommends specific manufacturing approaches to kind of maximize that probability of success. So that model is revised over time as we collect more data, as we change our processes. But there have been dramatic improvements in methods, artificial intelligence methods in general, to predict protein structure from amino acid sequence. So that problem kind of saw only incremental improvement over the last 20 years. At the end of last year, there was a paper put out by DeepMind that showed really massive advancement in that capability. And models like that have really profound implications for biosecurity, but not necessarily as weak points. I think they can be strengths. So you'll often hear machine learning folks talk about running a model backward. And so that's to say you could have a model that maybe takes biological function as input and outputs a protein sequence that would fold into an enzyme to carry out that function, right? So you have a model that has function mapping to sequence. And in theory, at least, you can run that same model in reverse to take in a protein sequence and produce some estimate of the likely biological function of that sequence. That kind of predictive risk assessment would be a huge step forward for biosecurity in synthetic biology, because it would give us the ability to kind of estimate risk on totally novel sequences. And something like 20 to 30% of the sequences we synthesize now are very novel. They don't have homology to kind of anything in any of these reference databases. And so tools like this are going to be really important in helping us do risk evaluation.
Yeah, and I think that's really interesting, though, because as the space continues to evolve, there's also not just the customers that Twist works with and the government agencies, but also, your own internal company. I'm actually surprised to learn that AI was not a weakness. I was all like, oh my God, if an algorithm goes wrong, the DNA is going to get messed up and you get these wacky sequences. But if that's not the risk, then, are there internal risks that Twist does need to protect itself against?
Yeah, and it is really important that companies don't only see risks as external events, right? You have to also consider what you often hear of as insider threat risks. And so at Twist, I think the primary way we do that is there is no way to synthesize a gene and Twist without going through biosecurity screening. So whether it's our customers or our internal research and development groups, they all order sequences through the same interface. They all go through the same biosecurity screening process. We do follow up with our R&D folks if they order something that triggers some level of concern. So that's really the chief way that we try and reduce any risk of what we hope would be zero insider threat, but just in case, you have to have systems in place that sort of default back to safe.
Right. I mean, that makes a lot of sense to me, because it's like a trust but verify. Like, you want to be able to trust everybody, but the last thing you think you want to do is say, and everything's great, everything's peachy, and not do the follow-up. I think that's the part that we always want to not miss. But I'm also thinking about this in the context of innovation, because, in startup culture, it's kind of cutthroat. I mean, the drive to innovate is so intense. My concern would be, does biosecurity and biosafety, maybe not as much biosafety, but definitely biosecurity, kind of fall to the wayside? I mean, if you're trying to innovate so fast, are you not considering those risks? And how do you communicate a plan for biosecurity as a young startup?
And that is a real challenge. So I think Twist, since its founding, saw biosecurity in the same way that maybe finally a lot of startups are seeing cybersecurity investment. This is something you have to get correct right from the start. You have to make a core part of your business. These investments, so cybersecurity investments, I think there's general appreciation that they protect your system, your company's assets, protect your intellectual property. Biosecurity measures do the same thing. They protect your employees, your community, your industry as a whole. So any VC investing in biotech really should ensure that biosecurity is taken incredibly seriously. Ask potential founders, do you have a biosecurity plan? Have you thought through not just how your technology is going to change the world, because founders are great at that, but are there security implications associated that come along with this technology you have? And if so, how do you plan to mitigate those concerns right from the start?
No, I think that's huge. And I always kind of go back to the the Facebook and Twitter kind of analogy of huge platforms changed the world and the risk was not assessed. And here we all are, there's a huge, yeah, right? It's like,
yeah.
I just like the last thing I want to happen. And my biggest concern would be if these platforms that make DNA and all these powerful biological sequences don't do the risk assessment, all power, no responsibility. Oh my God, what a crisis.
And in some ways, like biotech's a little bit ahead of that because there's a long history in biotech of thinking about dual use risk, which is great. It's been fascinating to me to start to hear people talk about some of these platforms, information platforms, as dual use information platforms. Like that's fascinating to me. And absolutely they should be thinking through these risks.
What does that mean, dual use?
Dual use is a term that's used to describe a technology that has like great benefits, but also potential harmful downsides. And so a lot of biotech technologies are inherently dual use. But as we've seen, I think a lot of information technologies have dual use contours as well.
Yeah, I think the thing about being honest about it and being upfront about it, it's like, yeah, there are potential downsides, and this is how we are managing that risk.
That's right.
I think that's how we go forward. Yeah, exactly. So I want to kind of take a step back and look at what's next for biosecurity. We touched on it a little bit of, running the machine backwards in terms of AI models, but what are some of the new or the upcoming challenges that you're looking at as head of biosecurity? What's kind of coming down the pike here?
I think in the near term, there are two things we've been focused on. The first is lack of screening on pools of shorter DNA sequences. So a gene that you might think of as maybe 1,000 base pairs long, so 1,000 A's, G's, T's, and C's. Shorter sequences are maybe 100, 150. Those are referred to as oligonucleotides or oligos. And DNA synthesis companies make very large pools of oligos. We also use oligos to make genes. That's how we make genes. We print small pieces of DNA and we stitch them together. This 2010 guidance document from the US government that I mentioned earlier, it explicitly said there's really no need to screen any sequence less than 200 base pairs. But if a gene length sequence should be screened for biosecurity concerns, and you can make gene length sequence from an oligo pool, why shouldn't you be screening oligo pools? No. Right?
Yeah.
So right now we're working on, we have an in-house algorithm to do that. And the goal is really, we want to demonstrate that this is feasible. We want to show how to do so. And by doing that, hopefully we encourage more synthesis providers to start to screen these oligo pools, because otherwise it's a pretty big hole in the sort of safety net around biosecurity screening for DNA synthesis.
It's a huge hole. It's like, oh, well, the pieces we don't have to screen, but when you put the pieces together, we have to screen. And it's like, wait, no, you do have to look at the pieces. They're part of the whole. That to me is a little bit obvious.
Well, and the challenges around screening oligo pools are slightly different. They have a really high false positive alert rate, right? It's harder to do, which I think is where some of that guidance came from. But there are ways to do it, and we should make sure that happens, because there's also increasingly a lot of open source protocols for how to make genes from oligos. Lots of labs in the country are starting to make their own genes from oligo pools. And so as that gets more and more common, we really have to be more responsible in terms of how we produce and screen these oligo pools.
So for some of these labs that are now, putting together their own genes using these oligos, or even for benchtop printers now where people can print DNA, oligos, or genes in their own lab, what are some of the ways that biosecurity can be introduced into that system? I mean, it seems like you would need some protocols maybe so these devices don't print certain sequences. How does that even work? Or how could it work?
That's a real challenge. So these devices, there's several companies working on them. The goal is to put a little device in your lab that can make DNA on demand. And that's great, but you need to make sure that those devices also have kind of the same biosecurity screening capabilities as you might have from a centralized provider. And importantly, that those capabilities can't be circumvented. So, a good example here is if you have a Xerox machine or a LaserJet printer and you try and print a picture of a $20 bill, most of those devices will refuse to do it because there's a risk of using that in counterfeiting.
Oh, I had no idea.
And,
that's cool.
Right? It's very cool. So that's those device makers taking a lot of responsibility for misuse of their devices. The same thing happens to happen on the benchtop side, but it's a hard technical challenge. It's something the companies that make those devices are really working on, but you have to come up with a way where you maintain maybe the privacy of what you're sequencing or synthesizing, because that's what some of the companies prioritize, versus ensuring that biosecurity screening remains consistent. That's a real challenge.
Wow, that's a whole can of worms right there. And I'm glad we talked about it because, it's been kind of on my mind of like, well, as if it's not coming from a central provider who's checking every PO box and making sure it's not in North Korea, and then I know that a lot of benchtop pincher companies make sure they're not sending benchtops to PO boxes, but there's a lot going on. Yeah, right. Like, don't send a benchtop. Yeah.
Right. But importantly, I think the goal for the industry as a whole should be uniformity, because what you don't want is for someone who really wants to acquire something and misuse it, you don't want them to do what's called venue shopping. So just go and look for the provider who has the lowest bar to acquiring something and see that as an economic incentive to buy from that provider. You want instead to have everyone kind of have a uniform capability. And I think that's really hard to achieve.
Yeah. Is that a government regulation, or is that sort of like an industry collaboration?
So right now, there's a lot of industry collaboration going on in terms of these best practices. So Twist is a member of the International Gene Synthesis Consortium. It's an industry group dedicated, strictly to kind of helping DNA synthesis companies understand what the current best practices are and to try and encourage that uniformity of screening. They published a harmonized screening protocol and help companies sort of come to achieve that.
Yeah. I want to look at now even a bigger picture. Because this is my favorite question that I get to ask our guests, which is, what can we build with biology? But in this context, right, I want to ask, what does a biosecure future look like? What's a picture of that from your perspective?
Yeah, I think there are three important things that need to happen to get to that, biosecure future. The first is really monitoring and improving tools, data sets, algorithms, all the kind of nuts and bolts pieces that we use for estimating the security risk associated with biological constructs, and importantly, novel biological constructs, right? That's a real emerging challenge. So scientists are getting really good at designing these novel protein structures to accomplish some specific goal that may not exist in nature. And those methods will produce like really unnatural looking protein sequences, things that have no homology to anything in nature. And that means we need better tools for saying like, oh, this thing doesn't look like anything natural, but it could be used to do some certain biological harm to a living system. And this is a really hard problem, but we can't continue to rely on the current state of the art, which is really comparing everything to naturally occurring, well-characterized proteins as the only form of risk estimation. Like that can't continue.
I'm just thinking about the problem itself of like, it looks like nothing we've ever seen before. How do we know if it's a risk or not? That is a huge challenge just by itself. And then you said there were two more pieces. So,
and the two more pieces are sort of larger scale kind of community aspects. So the second, I think, would be you need to build a research and industrial culture that kind of understands how to think through security concerns, because that's not a skill set that really we teach necessarily in grad school. But the Engineering Biology Research Consortium, EBRC, with whom Twist does a lot of work, they have a DHS-funded program that they call Malice Analysis, which is a great name, that teaches researchers like PIs, postdocs, grad students, how to think through security risks in their own research. So look at a piece of research that they understand well, they work on every day, and try and look at it from a different view to say, could this be misused? And so if that program is successful, future generations of synthetic biologists should develop this reflex to look at their discoveries and say, yes, it's wonderful, it changes the world, but also, could it be misused? And if it can, how can I work to reduce the risk of that misuse? So that cultural shift, I think, is the second priority. And third, I think you need really kind of a large-scale dialogue between synthetic biology companies and organizations, governments, civil society organizations, and kind of share these best practices and ensure that we're kind of addressing all the various stakeholders and risks posed by these technologies, and that we're all working together to kind of mitigate those risks. So DNA synthesis and screening is a really important part of that. But there are lots of levels in that supply chain. Think people that are designing organisms, designing pathways, who should all be thinking about the security implications of what they're doing. So that, better algorithms and tools, a mature scientific culture around security thinking, and a really engaged stakeholder community. I think that's the most effective way that we could build this biosecure future.
Wow. I mean, the cultural aspect to me is so interesting. It's almost like teaching bioethics needs to now go hand in hand with teaching biosecurity, both of which I think are lacking. I think there's so much focus on engineering and the, understanding the biology itself, which is quite complex. There's this second piece
Wait, in bioscience grad programs, you generally have to take an ethics class, and that's extremely important. You want biological PhDs and postdocs coming out and understanding the ethical implications of what they're working on. And I think, as you say, that needs to be paired with also thinking about the security implications of their work. Both of those are incredibly important.
This is interesting that it comes in a grad school, but does this need to come also into undergrad? Do we need to be introducing bioethics and biosecurity into undergrad programs or even high school programs for biology? Would that help, do you think?
I mean, I'm certainly biased, but I don't think there's any age at which thinking through the ethical considerations of what you do and the security considerations of what you do isn't applicable. That should apply as broadly as possible. And that mouse analysis program I mentioned is branching out into industry. So it's important to engage with academics, absolutely. Also important to engage with industry so that industry understands that the technologies they're working on have security implications and that those should be thoughtfully addressed, in the product lifecycle.
This is so much to think about, James. Thank you so much for being on the show. Your work is really, really important. And so how can our audience stay in touch with you? How can they keep up with your work?
The best place is twistbioscience.com. I really appreciate the time.
That's our show, folks. Thanks so much for tuning in. This episode was edited and engineered by Bryce Joseph Nelson and produced by Nikita Michelson. I'm going to assume you're listening to this show on your favorite podcast platform right now. So head on over to the subscribe button, click it, and you'll get our next episode the second it drops. Until next time, dream big about what you want to build with biology.